In order to provide communication among the cells of the body, the billions of neurons in the nervous system must first communicate with one another or with effector cells via synapses. Synapses allow information to be filtered and integrated. Synapses are either electrical or chemical. Electrical synapses are called gap junctions and have many ion channels called connexons connecting two adjacent cells. Electrical synapses allow quicker communication, more coordinated communication between cells, and two-way transmission. A physical separation between adjacent cells, called the synaptic cleft, is characteristic of a chemical synapse. Because action potentials cannot cross a synaptic cleft, this electrical signal is converted into a chemical signal using neurotransmitters to cross the cleft. Unlike electrical synapses that allow for two-way transmission of signals, communication at chemical synapses is one-way only. At the presynaptic synapse, a series of events occur in order to communicate with the adjacent cells. When an action potential arrives at the presynaptic membrane, a depolarization phase opens voltage-gated calcium channels, allowing the inflow of calcium ions from the extracellular fluid. Increased calcium in the cytosol triggers exocytosis of synaptic vesicles, carrying neurotransmitter chemicals and releasing them into the synaptic cleft. These neurotransmitters then diffuse across the cleft and bind to receptors. Then the ligand-gated ion channels on the postsynaptic side will allow ions to flow according to their concentration gradient. Sodium entering the cell makes the interior slightly more positive, while potassium leaving the cell makes the interior slightly less positive. And then depending on which ions enter the postsynaptic cell through the channels, the ionic flow will cause either a graded depolarization or hyperpolarization in the postsynaptic membrane. Large, graded depolarization tend to generate action potentials. And if the depolarization is above threshold voltage, then action potential will be generated. The neurotransmitters bind to a specific ligand-gated ion channels in the postsynaptic membrane. A graded depolarization or hyperpolarization will occur depending upon which channels open to ionic flow. Depolarizations resulting of the sodium gates opening stimulate the generation of action potentials. These depolarizations are known as excitatory postsynaptic potentials, or EPSPs. Hyperpolarizations result of chlorine and potassium gates opening inhibit the generation of action potentials. These hyperpolarizations are known as inhibitory postsynaptic membrane potentials, or IPSPs. The sum of all IPSPs and EPSPs from all synapses determine whether the action potential will be generated at the neuron's trigger zone. Let us explore that. A typical neuron may have 1,000 synapses. The neuron will have a corresponding number of postsynaptic membrane potentials summed at its trigger zone. Spatial summation results from the effect of potentials coming from many different synapses, while the temporal summation results from the effect of potentials coming from one location or synapse, but many times in succession. Summation can result in three possible scenarios: IPSPs, and EPSPs may summate and result in hyperpolarization of the membrane. In this case, no action potential will be generated, or IPSPs and EPSPs may summate, resulting in a subthreshold depolarization of the membrane. Again, no action potential can be generated. Finally, IPSPs and EPSPs summate, resulting in an above threshold depolarization. This will generate a new action potential.